Hello, I am Charlotte DiBiase. Thank you for viewing my presentation on maternal floral color, UV protection, and germination in Ipomopsis aggregata. Maternal effects are one of the ways by which adult plants can influence their offspring. They are defined as the contribution of the maternal parent to progeny phenotype beyond the equal chromosomal contribution of both parents. This means that via a physical connection, environmentally regulated chemicals circulating through a maternal plant are passed into developing embryos. As can be seen on this slide, as the seed is developing, structures such as the funiculus allow for maternal chemistry like hormones, nutrients, and pigments to diffuse into the embryo. In all plants, light between 680 and 700 nanometers is required for photosynthesis. Unfortunately, incoming sunlight also contains short wavelength, high energy UV radiation, which can significantly hinder a plant's photosynthetic capabilities by damaging proteins involved in the electron transport chain and the thylakoid membrane. Preliminary data collection shows how elevation affects UV levels, with higher elevated locations experiencing more UV exposure. This can be seen in the bar graph on this slide, which shows how the 2,000-foot elevation gain between the city of Colorado Springs and Manitou Experimental Forest caused a drastic increase in UV exposure. From this information, we can infer that Manitou Experimental Forest, located at approximately 8,000 feet, experiences high levels of UV radiation. This UV stress has led to adaptations in Rocky Mountain native plants hoping to maximize fitness. Scarlet gilia, here Ipomopsis aggregata subspecies Colina, is a member of the Polemoniaceae family. It is a perennial herb native to the American West and lives from 1 to 8 years as a basal rosette, then bolts and flowers once before senescence. Across the Front Range of the Rocky Mountains, an interesting spectrum of floral color morphology exists within the species. Flowers have deep corolla tubes as they are pollinated by long-tongued hummingbirds and hawk moths. The color of petals ranges from lighter pink to deep scarlet thanks to the red secondary flavonoid pigment anthocyanin. Because of its antioxidant and pigment properties, anthocyanin has been shown to decrease UV stress in plants by mitigating oxidative damage in high UV conditions, as well as by physically shielding chloroplasts from high wavelength radiation. Previous work on the species postulates differentiation between light and dark red floral morphologies, either in response to difference in pollinator preference or as adaptation to UV stress. As can be seen in the bar graph on this slide, adult Ipomopsis aggregata plants in MEF have shown significant differences in photosystem efficiency between dark red and pink individuals, with darker morphologies having an advantage over 3 consecutive field seasons. Darkly colored adult plants also had significantly higher anthocyanin and chlorophyll content, indicating that dark color is due to high anthocyanin content and could be related to advantages in chlorophyll content. Thus, previous field data has led to the hypothesis that anthocyanin content influences stress tolerance mechanisms in order to provide advantages to individuals with darkly colored petals. The advantages anthocyanin provides to adult plants suggests other life history advantages of dark floral color. In this study, I aim to address the influence of maternal floral color on reproduction and realized fitness through the metrics of seed number, seed biomass, germination rate, and germination success. I also aim to address the effects of anthocyanin content in the seedling stage of Ipomopsis life history through functional traits including photosynthetic potential, biomass, anthocyanin content, and chlorophyll content. During previous field seasons, 12 dark and 12 light adult plants were collected from 3 sites in Manitou Experimental Forest, and their seeds were harvested. The 3 sites in MEF had similar soil moisture profiles and were comprised of meadows surrounded by ponderosa pine and aspen trees. For this experiment, seeds from each plant were counted and weighed to determine maternal effects on reproductive efforts. Then, seeds from each adult plant were randomly selected and placed onto agar plates in a growth chamber. Growth chamber conditions were set according to average temperature and photoperiod of MEF and surrounding areas. Germination was censused for 21 days, and after the final census, 6 seedlings were chosen from each maternal plant. Physiological measurements were taken to determine the effects of maternal floral color on seedling life history. In this figure, parts A and C show the effects of dark and light red floral color on seed number and mass. While part A shows there was no significant effect of maternal floral color on seed number, part C shows there was a significant difference in the mass of seeds produced by light and dark red maternal plants. More massive seeds from dark red adults suggests that anthocyanin content is positively correlated with reproductive effort. As was expected, parts B and D show there was no effect of site within Manitou experimental forest on seed number or biomass, suggesting that the forest's population is not varying in reproductive strategy according to microenvironment. Results of the germination assay show a significant difference in germination success between seeds from dark and light red maternal plants. While initial germination status was similar, more seeds from dark red adults ultimately germinated. Seeds from dark red adults also germinated at a greater rate according to Wilcoxon survival analysis. The greater germination success of seedlings from dark maternal plants suggests that darkly colored adult plants are able to provide seeds and thus seedlings with growth advantages. Seed biomass and germination success translate to physiological advantages in seedlings from dark red maternal plants. In this figure, part D confirms that dark red maternal plants are somehow passing their high anthocyanin content down to their progeny. This could be by genetic contribution, or it could be via maternal effects. Part A shows how dark-derived seedlings were significantly more massive, suggesting that the more massive seeds produced by dark red adults are able to grow into larger seedlings. Part B shows how photosystem efficiency was also significantly greater in dark-derived seedlings, which is concurrent with dark adult plants being more photosynthetically efficient in previous field seasons. This finding implies an intergenerational efficiency advantage of plants with dark red floral morphology. Chlorophyll content index, shown here in part C, was not significantly different between seeds from dark red and light pink maternal plants. This is not concurrent with data from dark and light adult plants, however, likely indicates a difference in the way anthocyanin interacts with chlorophyll content between adults and seedlings, rather than anthocyanin lacking influence during the seedling phase. Because seedlings had not experienced the continuous UV stress their maternal plants did, anthocyanin had no UV exposure to protect chlorophyll content from, and thus did not affect chlorophyll content. Realized fitness in Ipomopsis aggregata is affected by floral color morphology and thus anthocyanin content. Dark maternal plants were able to produce larger seedlings which germinated faster and at a greater proportion than seeds from light maternal plants. Maternal effects could be the mechanism by which adult plants are able to influence their progeny. Maternal plants experiencing high UV levels could upregulate production of anthocyanin in order to minimize UV damage. This particularly high concentration of anthocyanin in dark red maternal plants could then be transferred into seeds during development, producing both seeds and seedlings with higher anthocyanin content. The higher photosystem efficiency of darkly colored adult individuals could also provide maternal plants with more photosynthate to allocate towards reproduction, likely leading to larger seeds with more nutritive endosperm. More massive and photosynthetically efficient seeds from dark maternal plants further delineate how darkly colored adults are able to maximize fitness using UV stress tolerance mechanisms. Given these findings, it is likely that anthocyanin plays a large role in plant adaptation to UV stress and floral morphology. The influence of maternal effects and their ability to produce a morphologically diverse population of individuals within Ipomopsis aggregata at MEF suggests much larger implications for how anthocyanin content and UV stress shape the species' evolutionary history. I would like to thank Shane Heschel and Mark Snyder for their help advising me on this research, as well as Rob Heavey for funding it. I would also like to thank Alex Shapiro for her help collecting field data and on germination censusing. My project would not have been possible if it weren't for all of the previous research performed by alumni of the Heschel Lab. Lastly, thanks to Donna Sison and the OBE department for their continued support throughout my time at CC. Thank you.